Hi everyone, welcome back to my educational channel on biology. This is part 2 of experiments on photosynthesis. So in this video, we're going to discuss effects of carbon dioxide concentration and temperature and also color of light on the rate of photosynthesis. So we discussed the experiments separately, and in the first video, I've already discussed investigating the effects of light intensity on the rate of photosynthesis. If you have not viewed that video, please do view it, all right, because it is very well planned and systematic, and a lot of questions are given to test you and also explain in depth. Okay, there's an explanation, in-depth explanation of the technique of answering for each question. It will help you to score an exam. Likewise for this video too. Okay, so we will be discussing paper 3 answering techniques, and I will try to teach you as best as I can based on my vast experience in conducting practical tests and also in marking. Right, so let's get started. So just to recap from my very short video, I've already explained that one technique of preparing the Hydrilla species so that it can release air bubbles that can be counted is to use this method, okay? That is recommended on by many teachers, by a few teachers on YouTube, okay? The teachers from other countries. Now, watch a very good video on the effect of light intensity on rate of photosynthesis at this link, okay, which I'll show below. At 1 minute 30 seconds, you'll see how to cut the stem. There's a technique here. And the important technique is to cut the top end of the plant, the Hydrilla plant, at a slant inside the sodium bicarbonate or sodium hydrogen carbonate solution. It's the same word, same meaning. So inside the sodium hydrogen carbonate solution, not outside. If it's outside, then air from outside may get in. Okay. So first you turn the plant upside down. This is what they do in the practicals. Okay, for those from other countries that teach the GCSE level syllabus. So turn the plant upside down first. Okay. So this is the bottom end of actually. Now this is called the upper end already. So you cut using a scissors and in the water or in the solution, you cut the plant so that there is a fresh cut here, and then the holes are open up. Okay, the vessels are open up so that it can release the oxygen bubbles. Okay, and cut the stem inside the solution, and the slanted cut will help the surface area to be a bit more, so a bigger bubble can be released. Okay, so cut in water so that this prevents the air outside from blocking the cut end, and then that will stop the bubbles from being released, the oxygen bubbles from being released. So after you cut the end, you wait a while, let the plant be exposed to light, and it should slowly produce the air bubbles. Okay. Now this technique can be used. First you must follow your paper, the question paper. What instructions are given? All right. And if they have their own way of doing it, then you follow the way that you're instructed to do. I don't know what the question will be like, right? So it's up to you to decide what to do. But if as a final, if you have done everything and bubbles cannot be released somehow, all right, then you can try this technique, okay, which is very fast. Turn the plant upside down, cut in a slanted manner, and then watch the bubbles release from the stem, and count all these bubbles released from the stem. Not the tiny bubbles on the leaves. This one they stuck to the leaf, and some don't float up. Float out also very tiny only. So there may be a few of them. It's the big one from the cut part of the stem that we count, the big bubbles. So again, some background knowledge of photosynthesis. The word equation for photosynthesis is simplified as wet go. W for water, E for energy, which is light energy, C for carbon dioxide, another C is for chlorophyll. WECC, and then arrow GO, go are the products, glucose and G for glucose and O for oxygen. Now, what are the factors that affect the rate of photosynthesis, and what is the theory behind it? What's the biological concept behind this? So for light intensity, the light is needed to produce or to provide the energy. Light provides the energy for the photolysis of water, which is the splitting of water into the hydrogen ions and the hydroxy ions during photosynthesis. Okay, so light is needed. So as the light intensity increases, the rate of photosynthesis will increase until a certain value, okay, a maximum value. Now, carbon dioxide. Carbon dioxide is the substrate, is an important substrate for photosynthesis. So it's needed in photosynthesis, right? So if you increase the carbon dioxide concentration, against the same graph as for light intensity, so as we increase the carbon dioxide concentration here, the rate of photosynthesis will increase because there's more carbon dioxide which acts as a substrate. In fact, the carbon dioxide is reduced in the light independent reaction, LIR, to produce glucose and oxygen as a byproduct. Okay, oxygen is a byproduct. So the higher the concentration of carbon dioxide, then the higher the rate of photosynthesis. Now for temperature affects the photosynthetic enzymes that are involved in photosynthesis to catalyze the process of photosynthesis. So the optimum temperature for the photosynthetic enzymes to act is 28 or 25 to 30 degrees Celsius, lower than for humans. And at high temperatures like 50 or 60 degrees Celsius, the enzymes are denatured and their structure, the protein structure, is destroyed. Okay, then active sites are also destroyed. The protein structure is changed, all right, and then the active sites are destroyed. So at high temperatures, the rate of photosynthesis will decrease down to 0. Now, color of light also affects the rate of photosynthesis because red and blue light are well absorbed by the chlorophyll and other pigments in the plant, so they can be used to provide the energy for photolysis of water. But the green light is not absorbed by the chlorophyll, it's reflected. So if a plant is placed in green light, the rate of photosynthesis will decrease, okay, because the green light is reflected from the plant and the plant will absorb less light energy for photosynthesis. Now, investigating effects of carbon dioxide concentration on the rate of photosynthesis. So there are two manipulated variables and two responding variables. I call them the main variables and sub-variables. So for MV, we have the main MV and the sub-MV. And for RV, we have the main RV and the sub-RV. So let's talk about the main MV and the main RV. These are obtained from the aim of the experiment, okay, to investigate effects of carbon dioxide concentration on rate of photosynthesis. So the carbon dioxide concentration is the main MV. It's taken from the aim or objective. And the rate of photosynthesis is your main RV, okay? Now, so effect of something, that one is the MV, effect of MV on RV. How about the sub-variables? Sub-variables are those used in the experiment, in the laboratory. For example, how do you change the concentration of carbon dioxide? You change the concentration of the sodium hydrogen carbonate solution, right? Because the sodium hydrogen carbonate solution provides the carbon dioxide from the carbonate group here, right? So the higher the concentration of the sodium hydrogen carbonate solution, then the higher the carbon dioxide concentration, because more carbon dioxide will be provided to the plant. Okay, so this is your sub MV, which when it changes, it will change the, when the value changes, it will change the carbon dioxide concentration. Now, for the responding variable, what is the sub RV? That means, how do we measure the rate of photosynthesis? So we count the number of air or gas bubbles, oxygen bubbles, released in either 1 minute, 2 minutes, or 5 minutes. Okay. Now here you must read the exam question carefully and underline in how many minutes. You're supposed to count the air bubbles in how many minutes. That's released in how many minutes. Okay. So it may not be the same like the textbook. Textbooks is counting the number of, recommends counting the number of gas bubbles released in 5 minutes. Okay, it may be a shorter time because the practical is quite short. Now, next, so the sub-RV is used to calculate the rate of photosynthesis. So rate of photosynthesis is actually the average number of gas bubbles or air bubbles per time. Per time. So if your number of air bubbles or gas bubbles released is for 5 minutes, then you have to take that number divided by 5 minutes to get your rate of photosynthesis. Okay, so in this picture, you see the sub-MV is the concentration of the sodium hydrogen carbonate solution, and the sub-RV is the number of air or gas bubbles released in, say, 5 minutes. Okay, and also one thing to note is we must write the term sodium hydrogen carbonate solution. Don't just write sodium hydrogen carbonate. Some students make this mistake. You forgot the word solution. The word solution is important, okay, because this this compound is in the form of a solution. Let's now discuss this experiment based on the textbook, investigating the effects of carbon dioxide concentration on the rate of photosynthesis. Our aim to investigate the effects of carbon dioxide concentration on the rate of photosynthesis. So the effects of something that is the MV, okay, the effects of MV on RV. So carbon dioxide concentration is our main manipulated variable that is taken from the aim, and the rate of photosynthesis is the main responding variable, or RV. Now, a hypothesis. Hypothesis consists of the main MV, main RV, and the correct relationship. So the higher the carbon dioxide concentration, the higher the rate of photosynthesis. So MV, carbon dioxide concentration, RV is the rate of photosynthesis, and our relationship is the higher, okay? Now, why is carbon dioxide needed, and how does it increase the rate of photosynthesis? As we have discussed, it's because carbon dioxide is a substrate for the process of photosynthesis, and the carbon dioxide is reduced to form glucose during the process of photosynthesis. Now, variables. Our manipulated variable is, there are two, the concentration of sodium hydrogen carbonate solution. The sodium hydrogen carbonate solution produces or provides the carbon dioxide for photosynthesis to the plant, right? So it is the sub MV, because it will be used to change the carbon dioxide concentration, okay? So this variable is changed to change the carbon dioxide concentration, okay, when doing the experiment. So carbon dioxide concentration remains as the main MV, and the sub MV is the concentration of sodium hydrogen carbonate solution. Please don't forget this word solution. Some students miss out this word, so it's not correct without this word solution. Responding variable. The number of air bubbles released in 5 minutes is our sub RV, and the rate of photosynthesis remains as the main RV. So to calculate or determine the rate of photosynthesis, we have to count them and record the number of air bubbles released in 5 minutes. Now, it can be 5 minutes or 2 minutes or 1 minute, depending on the question. Please read the exam question carefully, okay, and underline how many minutes. So the number of air bubbles released in 5 minutes is called the sub RV, and from it we can determine the rate of photosynthesis, which is the main RV. Okay, so sub RV is used in the experiment that we carry out in the lab. All right. Now, constant variables. The distance of light source from Hydrilla species. This is a good constant variable because there is a value, okay, to it, and we can write this constant variable and then mention how to handle it, okay, if we are asked. The type and size of Hydrilla species. Size means like, how many cm of Hydrilla species stem did you cut? And also type, because Hydrilla species got many types of species of this Hydrilla. Must choose the same species. The voltage of the bulb. Now, materials. Hydrilla species, distilled water, sodium hydrogen carbonate solutions of different concentrations, such as 0.1 m, 0.2 m, 0.3 m, and so forth until 0.10 m. Now, these are many solutions, right? So normally in exam, maybe for your lab exercise, you may pick 4 or 5, okay, concentrations of sodium hydrogen carbonate solutions. You won't be able to do all in a short time to test all of them. So, but in our practical exam, probably they may ask you to determine the number of air bubbles released in 5 minutes, maybe from 3 solutions only, because there's lack of time and you have to answer the questions. Now, apparatus: scissors, 60-watt bulb, meter rule, stopwatch, paper clips, boiling tube, measuring cylinder, retort stand and clamp, thermometer, and beaker. So let's look at the method to set up this apparatus for this experiment to investigate effects of carbon dioxide concentration on the rate of photosynthesis. First, let's start with the Hydrilla species. So attach a paper clip to one end of the stem, okay, stem cutting, which is about maybe 8 cm or 6 cm of the Hydrilla species plant. Okay, read the instructions carefully and find out what you need to do, how much Hydrilla species, what size of Hydrilla species plant you need to cut. And then you use the paper clip to weigh down so that the plant can be fully immersed in the solution and it won't float upwards. If it floats upwards, then you cannot count the number of air bubbles or gas bubbles. So this is a precaution, okay? Put the paper clip at the end. Now, using a measuring cylinder, measure and pour out 5 ml of 0.1 m sodium hydrogen carbonate solution and fill it in the boiling tube. Put it in the boiling tube here. Next, put the plant in the boiling tube itself. Okay, put the plant inside, and then you clamp the boiling tube vertically, okay, so that it is stable, and the plant is fully immersed in the sodium hydrogen carbonate solution. So next, place the bulb light a distance of 10 cm from Hydrilla species. So use a ruler to measure from the Hydrilla plant here to the filament, okay, that gives out the light. So that should be 10 cm. This is the fixed variable, okay. Then switch on the 60 watt bulb here, light, and then you wait 1 to 2 minutes for Hydrilla species to stabilize here and adapt to the new conditions. Then when you see the bubbles coming out, I know that it's starting to adapt. And if you are ready, then start the stopwatch. Okay, don't take too long for this because time is short. So time is short to do the experiment, so you must always be time conscious. So once you see the bubbles coming out, then when it's ready, start the stopwatch and count and record the number of air bubbles released in 5 minutes. This is your RV, sub-RV. Then take 2 or 3 readings and to obtain an average so that your result is more accurate, okay, to get more accurate results. Okay, then repeat the steps for other concentrations of sodium hydrogen carbonate solution, such as the 0.02 m, 0.02 m, 0.04 m, 0.06 m, 0.08 m, and so forth. Now, in the practical exam, they won't ask you to find out the number of gas bubbles released for so many MVs, so many value of MVs, maybe 3, all right, because there's lack of time, maybe 3 MVs, 3 manipulated variable values. The next step is to read the procedure again and try to identify the precautions, the MV, the RV, and the CV here, okay? So attach a paper clip to one end of a stem cutting of Hydrilla species to weigh it down so they can be fully immersed in solution. But first you have to cut the Hydrilla species a certain length, like 6 cm or 8 cm. Now, using a measuring cylinder, measure and pour out 5 ml of 0.1 m sodium hydrogen carbonate solution into the boiling tube. By the way, the first step is called the precaution, okay, because you put the paper clip so that it's weighed down, then you can easily immerse it in the solution and you can count the number of gas bubbles released, okay, it's a precaution. Now, after that, you measure out 5 ml of 0.01 m sodium hydrogen carbonate solution. So this is your MV, the first MV, 0.01 m, 0.01 m sodium hydrogen carbonate solution. Okay, so the concentration of the sodium hydrogen carbonate solution is your manipulated variable in the lab. Okay, the sub-MV. Now next, put the plant in the boiling tube together with the sodium hydrogen carbonate solution, and then clamp it vertically to the retort stem. And next, you have to measure a distance of 10 cm between the bulb light and the Hydrilla species plant. Okay, then you switch on the 60 watt bulb light, placed at a distance of 10 cm from Hydrilla species plant. Now, don't start the experiment immediately. Allow 1 to 2 minutes for Hydrilla species to stabilize and adapt to the new conditions, the new light intensity. As the bubbles start to be released, then you start the stopwatch, okay, when you're ready. So after each concentration of sodium hydrogen carbonate solution, you have to do the same thing. Number 5 has to be practiced. Okay, allow 1 or 2 minutes for Hydrilla species to stabilize. Next, count and record the number of air bubbles released in 5 minutes. Okay, so once you start the stopwatch, take note of the time, count and record the number of air bubbles released in 5 minutes from the Hydrilla species plant, especially from the stem. Take 2 or 3 readings to obtain an average value so that it's accurate. The readings are more accurate. The results are more accurate. Repeat steps 2 to 6 for other concentrations of sodium hydrogen carbonate solution, such as 0.02 m and so forth. Okay. Now this is your MV, the sub-MV that's changed, and the sub-RV that's a responding variable which will change according to the sub-MV is to count the number of air bubbles released in 5 minutes from Hydrilla species. Okay. Now when you write the word Hydrilla species in the exam, It's either italics or you underline the word Hydrilla. Underline, that is the nomenclature or the rules, okay, for naming the species of organism. So now let's discuss the paper 3 popular questions for paper 3 and the complete answers. Take note also that I will teach you the answering techniques. You can jot it down if you want, and also I will also tell you the mistakes that many students make so that you can avoid them, okay, in order to score high marks for your paper 3. Now, so these are our results, okay. Concentration of sodium hydrogen carbonate solution, this is your sub MV, is on the left hand side, and here we have the sub RV, which is the number of air bubbles released in 5 minutes. There are 2 readings, and after that you get the average number of air bubbles released in 5 minutes. That means you have to add reading 1 and reading 2 and divide by 2, okay. And then you may be asked to calculate rate of photosynthesis. Sometimes it's there, sometimes it's not. This question, sometimes the question is asked, sometimes it's not. Now, how to calculate rate of photosynthesis? You divide the number of air bubbles, the average number of air bubbles here, divide it by the time. The time is 5 minutes, right? So that means in 5 minutes, 2 air bubbles or gas bubbles were released. So per minute, how many gas bubbles are released? It's 2 over 5. 2 bubbles divided by 5 minutes. Okay, so you will get 0.4. Right? So all the working is shown here. It's better to show the working, safer, all right, for the rate of photosynthesis if you are asked to calculate the rate. And take note of the units. The units for average number of air bubbles released in 5 minutes, there's no unit because it's really mentioned air bubbles. If you want to put a unit, it should be bubbles, not minutes, it's bubbles. 2 bubbles, 7 bubbles, 11 bubbles, and so forth. Okay? So don't be mistaken. Don't write minutes here, it's wrong. It should be bubbles or just leave it alone. This is also in the textbook. Okay, the table that's given doesn't have any units, right? But our rate of photosynthesis is the number of air bubbles, no units there, divided by time in minutes. So your unit should be per minute for rate of photosynthesis, okay? Because it's divided by the time. So for rate of photosynthesis, according to this equation, your unit is per minute, and you have to write it down in the table. Okay, you also have to write down the concentration of sodium hydrogen carbonate solution. The unit for it is molar here, but it can be percentage. So unit of concentration may be percentage. Find out from the question that's been asked. Okay, don't assume it's M. Could be percentage. So all right, so these are the results, and the results are for the sub MV of 0.01, 0.02, 0.04, 0.06, and 0.08. Now in the practical test, which is actually 45 minutes, but you have to minus 5 minutes for checking of apparatus. So you have 40 minutes left. In the practical test, that's 40 minutes with you answering questions also, required to answer questions. I don't think they will be able to ask you to carry out the experiment for all these concentrations. Maybe 2 or 3. Maybe 2 or 3 of these concentrations only, if it were to come out. So here, to answer the biology paper 3 questions, we have to have a set of results. So I've taken out 3 results here, okay, for sub MV or MV of 0.01 M, 0.02 M, and 0.04 M sodium hydrogen carbonate solution, okay, and these are the results. Now you notice that when the sodium hydrogen carbonate solution is lower in terms of its concentration, okay, when the concentration of sodium hydrogen carbonate solution is lower, the average number of air bubbles released is less, and the rate of photosynthesis is also less. But as the concentration increases, you can see that the average number of air bubbles released increases, right? And then the rate of photosynthesis also increases. Okay. Now let's look at the first question. Is on observation. Based on the table of results above, state one observation from this experiment. Can you tell me what are the 4 components of an observation? Yes, you must have the sub MV, the MV from the experiment, okay, from the table, and then you must have the value of the sub MV here, right? Then you must have the sub RV, which is your result that you obtain in the lab, plus the raw value, not this one, not the average value, but rather this one. This is your sub RV that you have to write in. okay, the raw data or raw data, not the average number, because for average number of bubbles, you have already added both values, get the total, divide by 2. So this is no longer the observation. This one here is the observation, okay? So please don't make that mistake, as some teachers may ask you to write the average number. It's not correct. It should be the raw data, the number of air bubbles released in 5 minutes. Okay, try and write your own observation. Practice writing. Right, let's look at the answer. When the concentration of sodium hydrogen carbonate solution here, I choose the 0.041. So you look carefully what they want you to write. If they don't give you any particular concentration to write about, then you can pick your own. So either choose the lowest concentration or the highest concentration. Okay. When the concentration of sodium hydrogen carbonate solution is 0.04 m, the number of air bubbles released in 5 minutes is, okay, let's look at it, 10 for the first reading or result, and 12 for the second reading or second result. Okay, not 11, because you write 11 is wrong. This is not the raw data. Okay, it's not your observation. You did some calculation already, okay, to get this. So let's see, concentration of sodium hydrogen carbonate solution, sub MV. The value of the sub MV is 0.04 m. Okay, number of air bubbles released in 5 minutes is your sub RV. Okay, it's from the table. It's 10 for the first reading, and then 12 for the second reading. So you will get the full marks, which is normally 2 marks or 3 marks. Okay, so could be 1 mark for the MV, 1 mark for the RV, but together with the values. So a common question is to state the 3 variables and the methods to handle them. Okay, you may be asked 1 or 2 of these variables. So try to write down the manipulated variables and other variables, and also the method to handle them on your own first, okay, as a revision. Let's look at the answer. Now, for MV, the manipulated variable, is the concentration of sodium hydrogen carbonate solution. Can we put the concentration of carbon dioxide? It's not wrong, but in this experiment, we write down the sub variables, okay, that are used in the experiment. Is better. Okay, so concentration of sodium hydrogen carbonate solution is a better answer. So method to handle it is to use the sodium hydrogen carbonate solutions of different concentrations, such as, now you must take all the values that are used, okay, all the different MVs that are used. And the important word here is use something of different. Or you can say use different sodium hydrogen carbonate solutions of different concentrations. Okay, I add the word use here. Use different sodium hydrogen carbonate solutions of different concentrations, and then state the values. All of them must be done. All right, if you don't have these values, then you still get it wrong. So don't write the incomplete answer. Okay, you won't get the marks. Secondly, what's the responding variable? The responding variable is always the same, whether the factor is light, concentration of carbon dioxide, temperature that you are testing. Okay, is the number of air bubbles or gas bubbles. It depends on what they write in the test paper, or they could put oxygen bubbles also. So what they write there, you copy down, right? Don't write your own type of bubble. So number of air or gas bubbles, depending on the question, released in 5 minutes, right? Now mistake here is many students write number of gas bubbles released. They don't state the time, so it's not complete again. If it's not complete, you will not get the marks. Okay, so make sure you write in 5 minutes, complete answers. Count and record the number of air or gas bubbles released in 5 minutes using a stopwatch. This is the method to handle the RV. Take note, you must have the word count and record. Record is also very important. And not measure, it's not measuring, it's counting and recording. Okay, so count and record the number of air bubbles released in 5 minutes, and you must take the apparatus used. Okay, so this is another mistake. Some students forgot to write by using a stopwatch. Okay, so write complete answers and be careful to write complete answers. Thirdly, fixed variable is the distance of the light source from Hydrilla species. Why do we choose this variable? Because you can state the value very easily. It's 10 cm as stated in the instructions or procedure. Okay, so when you are reading the instructions in the question paper, select the fixed variable or constant variable that is that has a value, easy to write down. So our method to handle is to fix, we use the word fix, fix the distance of the light source from Hydrilla species at what value? 10 cm. Okay, sure to get the full marks. So the hypothesis for this experiment, what is it? Hypothesis, as we said, contains the main MV, mean RV, and relationship. We discussed that earlier. So the higher the carbon dioxide concentration, the higher the rate of photosynthesis. This was our hypothesis at the beginning. And here the relationship is higher, and our MV is the carbon dioxide concentration. Our main RV is the rate of photosynthesis. Next, let's go on to an important question that some students don't know how to write the answer. Based on this experiment, state the operation definition for photosynthesis. All right. Now, based on this experiment, so not on the last experiment about light intensity already. This is about carbon dioxide concentration, the effects of carbon dioxide concentration. So you must state the sub RV and sub MV from this experiment. And we use the tip. Now you can remember this tip. Dorem. Think of the word Doraemon. Dorem. So D for definition, the basic definition must be there. O for organism, any organism that's used. R is for sub RV. And then another R is for the RV, could be the main RV because that's stated here, photosynthesis. Okay, it doesn't matter. And then sub MV. Okay, let's read the answer. Photosynthesis is the process in which Hydrilla species produces organic food substances. This is the basic definition from carbon dioxide, and it is shown by the number of air or gas bubbles released in 5 minutes. So this is your sub RV. It's shown by the sub RV. Okay. Then the rate of photosynthesis is affected by the concentration of sodium hydrogen carbonate solution. So affected by. Okay. The RV affected by MV. And that's another statement. Okay. The RV affected by the MV. Right, now we go on to inference. State the inference for the observation in 1 about when the concentration of sodium hydrogen carbonate is 0.04 m. Okay, so what's the inference? So you have to note first whether it's the result, the number of air bubbles is comparatively higher or lower in the whole set of experiments. Higher, right? The number of air bubbles or gas bubbles released. Okay, so why is it that at that concentration, the number of gas bubbles released is higher than the rest? Or greater than the rest? Okay, let's look at the answer. Okay, try to write your own answer first, and then let's look at the answer here. So you have to connect the sub MV, which is your concentration of the sodium hydrogen carbonate solution, with the sub RV. Why is it that the number of gas bubbles is more, okay, compared to the other concentrations? That means you have to mention systematically. First, the sub MV affects the main MV. As you will see here, when the concentration of sodium hydrogen carbonate solution is higher, okay, it's 0.04 m, is comparatively higher compared to the rest, the concentration of carbon dioxide, concentration of carbon dioxide also becomes higher, because the sodium hydrogen carbonate provides the carbon dioxide for photosynthesis, right? So when the concentration of the carbon dioxide becomes higher, so you mentioned the main MV, how sub MV affects main MV, then you have to explain the biological effect of the main MV on the main RV. Okay, so this will cause the higher concentration of carbon dioxide, which is the main MV, causes more carbon dioxide to be reduced to form glucose and to produce oxygen as a byproduct during photosynthesis. Okay, so more carbon dioxide to be reduced to form glucose in the light independent reaction. So mentioned a little bit as the biological explanation, right? In that way, the rate of photosynthesis increases. So we have arrived at the main RV. The main MV affects the main RV through that biological effect. Because carbon dioxide is needed, is reduced to form glucose, and oxygen is a byproduct. Okay, so when the main RV, the rate of photosynthesis increases, this will cause more oxygen gas bubbles or more gas bubbles to be released in 5 minutes due to the higher rate of photosynthesis, and the oxygen gas is released as a byproduct. Okay, so now you can see the connection of sub MV affects main MV, MV affects main RV, and here to explain how it affects main RV, just a short sentence, and then main RV will affect the sub RV. Okay, so that's the secret of this inference here. So now we come to the drawing of the table of results. All right, so follow the instructions given in the question paper. They will probably tell you what the titles are. So don't forget the units. Okay, the titles could be concentration of sodium hydrogen carbonate solution as the sub MV, number of air bubbles released in 5 minutes, this is the sub RB, and then the average number of air bubbles released in 5 minutes, and they may ask you to calculate the rate of photosynthesis. So to make it realistic, we'll just take the readings. We'll just concentrate on 0.01, 0.02 m, and 0.04 m. All right, the rest we will not discuss, but it's the same pattern, same result. What I mean by same result is that it has the same relationship here, or same comparison. Okay. Yeah, so you're going to discuss three values, and we have already discussed how to calculate the rate of photosynthesis, and also stated that the unit may be percentage here, depending on the question. Now, for the graph, you may be asked to draw a line graph or a bar chart, right? In this case. So line graph, how to draw it? So normally, they will maybe ask you to find the average number of air bubbles released in 5 minutes for maybe about 2 or 3 values of MV only. Okay, not more than that, probably. Okay, so if they ask you to plot a graph of the average number of air bubbles released in 5 minutes against the concentration of sodium hydrogen carbonate solution, then the average number of air bubbles against something, that one is at the top, is the vertical axis, average number of air bubbles against something, that one is the horizontal axis. So the MV is on the horizontal axis and the RV is on the vertical axis. Okay, so first you must write down the title completely. Average number of air bubbles released in 5 minutes. Make sure you write down everything and not just average number of air bubbles. Okay, these words released in 5 minutes must be added. Okay, common mistake among students is that their RV or MV is not complete. And then also any units, if there are any units, you must write it down. For average number of air bubbles released in 5 minutes, there are no units because already told number is a number of air bubbles. So the unit is actually bubbles. But in the textbook also, they never give any units for this. They didn't give any units. But concentration of sodium hydrogen carbonate solution, there's a unit. It's either molar M or M molar or percentage, depending on the question. So take note of what the unit is in the question. Okay. Now next, draw the vertical line to represent the vertical axis and then the horizontal line. And then look at the values. For the MV is from 0.01 to 0.04. So your scale must be large enough to cover at least 3/4 of the paper. Okay. So these two boxes represent 0.01, 0.02 for another two boxes. So the scale must be regular. Okay. Then for the vertical axis is the same. It's from 2 to 11. So try to stretch up the scale until the end here to cover 0 to 11. Okay. All right. So scale must also be uniform here. Next, we are going to mark the first one is the axis, titles, units, uniform, scale. If that's all there, then you get one tick. Okay. Now, second tick or mark is for the points that must be correctly plotted. So looking at the points is 2, 7, and 11. So 2, 7, and then 11. Okay, according to the concentrations of sodium hydrogen carbonate solution. So if points are correctly plotted, you get one more tick. And the last one is your line graph. If it's a line graph, join all the points by hand, not with a ruler. Okay, for the graph. And certainly not the line of best fit, because for bio, you have to join all the points. So it may form a curve like that. So make your hand steady and then slowly draw the line. Cannot use a ruler to draw this line and this line. It's wrong. Cannot use ruler. So if it's a curve, draw the line freehand, smoothly to form a smooth curve. If your curve is smooth, then it's okay, you get the mark. Okay. Now if it's a straight line, then you can use a ruler. Okay, but normally we cannot get the straight line. But if it's a straight line, you can use a ruler. So for bar chart is another story. So now we have all the marks already. 3 marks for the graph. Okay, graph is 3 or 2 marks. Now for bar chart, it's different. But again, the axis, okay, must be the scale must be large enough. And let's see. Now in case, now read the question carefully. Maybe you're asked to plot the rate of photosynthesis against the concentration of sodium hydrogen carbonate solution. Okay, so we have calculated the rate here. And write down the type rate of photosynthesis. The unit is per minute, must be written down, or else you lose 1 mark. And for concentration of sodium hydrogen carbonate solution, of course, it's M, molar or percentage, depending on the question. Now, next, make sure that, okay, you see for the concentration of sodium hydrogen carbonate, it stands from 0 to 4, 0 to 0.0, 0 to 0.04. So make sure you use up all these spaces. You stretch out the values here. All I mean is that spread it out. So the first bar is here. Now see, there are regular intervals between the bars. Okay, that's one more thing to know. And the bars are of equal thickness. Okay, this is important. Now next, plot the points. All right, after you've written the titles, units, and have a uniform scale. Okay, there's one tick for you. And then the second tick is for the points. Make sure they're correctly plotted. Okay, 0. Now if you look at here, look at the table, 0.4, 1.4, and 2.2. Okay, 0.4, 1.4, and 2.2. Okay, then you draw the bar, okay, around here. Must be the same width, all right, and evenly separated out. Okay, evenly arranged here, but not stuck together. Don't join all the bars together. That will be a histogram. This is another common mistake made by students. Bar chart consists of discrete bars, separated bars, not joined together one. Make sure you know that. So if your bar chart has the correct shape and separate bars, then you get 1 mark. Okay, so 1 tick for the correctly plotted points, and then 1 tick for the shape of the bars, which are of the same width. Okay, and evenly arranged on this axis. All right. So read the question carefully. What you're supposed to plot against what. Okay, and write the whole title down. As I said, if you just write average number of air bubbles for this graph, is wrong. Okay, you lose 1 mark there. So let's look at this question on explaining the relationship between one variable and another, okay, based on the graph. So based on the graph, explain the relationship between the number of gas bubbles released in 5 minutes and the concentration of sodium hydrogen carbonate solution. Okay, so which one affects which one? Concentration of sodium hydrogen carbonate solution is your sub MV. It affects the sub RV, the number of gas bubbles released, okay, in 5 minutes. So start with the concentration of sodium hydrogen carbonate. As the concentration of sodium hydrogen carbonate increases, the number of gas bubbles released in 5 minutes increases. Note that this is like a hypothesis. So the relationship is like a hypothesis. When one variable increases, what happens with the other? All right. So when one increases, the other increases. Correct? Okay. What's the explanation? So relationship is 1 mark. Explanation may be 1 or 2 marks. Okay. All these questions are about 2 to 3 marks. All right. For observation, inference, and so forth. Now, explanation. The sodium hydrogen carbonate solution provides the carbon dioxide used as a substrate in photosynthesis. Therefore, as its concentration increases, as the concentration of sodium hydrogen carbonate solution increases, the concentration of carbon dioxide will also increase, right? Because the sodium hydrogen carbonate provides the carbon dioxide, releases the carbon dioxide for the plant to use. So then, if the concentration of carbon dioxide increases, the rate of photosynthesis also increases because the carbon dioxide is a substrate used in photosynthesis. Okay, so the carbon dioxide will be reduced to produce glucose, and then oxygen gas is a byproduct. Okay, so this will cause more oxygen bubbles to be released from the plant as a byproduct. Okay, so 1 mark for each tick, or they may combine together some of these answers. So explanation may be 2 marks. So here's a prediction question. The experiment is repeated with a distance of 30 cm between bulb and Hydrilla species using 0.04 m sodium hydrogen carbonate solution. Predict the number of gas bubbles released in 5 minutes and explain your answer. Usually for prediction question, they are trying to introduce or test you on another factor. Okay. So earlier we were discussing about concentration of carbon dioxide as a factor that affects the rate of photosynthesis. What do you think is the factor here that's being tested? Yes, it is the light intensity, okay? Because in the original experiment, you must compare to the original experiment, okay, that you did. In original experiment, the distance was 10 cm between bulb and hydrilla species. Now it's increased to 30 cm. So what happened to the light intensity? What happens to the light intensity? Will it increase or decrease? Yes, it will decrease because now the bulb is further away from the hydrilla species. So the light intensity decreases. And then it's using 0.04 m sodium hydrogen carbonate solution. So refer back for your to your table. When the sodium hydrogen carbonate solution had a concentration of 0.04 m at 10 cm, what was the value of the number of gas bubbles released in 5 minutes? Huh? You are not mistaken, it's 11 gas bubbles. So now you just change the distance to 30 cm, the light intensity is less. What will happen to the number of gas bubbles released in 5 minutes? You must mention whether it's less or more than 11. So should it be less or more? Yes, if you use the 0.4 m sodium hydrogen carbonate solution and 10 cm between bulb and hydrilla species, I use that as a comparison with this new experiment. Then you will see that the light intensity increases from 10 cm to the 30 cm distance. Okay, so the number of gas bubbles released in 5 minutes will be less. Okay, will be less than what? Than 11. Okay, than the original result. Okay, so the number of gas bubbles released in 5 minutes will be less than what? Less than 11, because 11 is the number of gas bubbles when the distance was 10 cm and the concentration of sodium hydrogen carbonate was 0.04 m. Okay, referring to the table, the original table of results. So now it will be less because now you have increased the light, you have decreased the light intensity. So explanation, so prediction is 1 mark, explanation may be 1 to 2 marks. As the distance between bulb and hydrilla species is increased, the light intensity decreases. Or you can just bracket this up, you can just write the light intensity decreases, okay, at a distance of 30 cm, okay, between bulb and hydrilla species. So less light energy will be absorbed by the plant and will be used for the photolysis of water in photosynthesis. Thus, the rate of photosynthesis decreases and there are less oxygen bubbles produced as a byproduct. Note that we have connected the variables again in the same sequence. First, we mentioned the sub-MV, which is now the light intensity. Okay, and sub-MV affects the main MV, which is the light energy. Okay, which is the light intensity. Okay, then the main MV affects the main RV. So the rate of photosynthesis decreases because there's less light energy for photolysis of water in photosynthesis. And then the main RV will affect the sub-RV. Less oxygen bubbles are produced as a byproduct. As we go on to see, the third experiment is investigating the effects of temperature on the rate of photosynthesis. Okay, so now our MV is temperature. Now for this part, I did not go into detail because it will be too long, but I just give you the rough idea here, and you can read from the textbook the experiment. Okay, the manipulated variable is the temperature. So method to handle is how to change the temperatures. It's still using the same setup, okay, of the Hydrilla species plant immersed in the sodium hydrogen carbonate solution. All right. So, but then how to change the temperature? And I can see here, the beaker outside acts as a water bath. Okay, so immerse the boiling tubes with the Hydrilla species inside in the water baths at different temperatures. Okay, such as 10 degrees Celsius. You may have to use a bit of ice to cool it down. 20 degrees Celsius, and then so forth until 60 degrees Celsius. Okay, it will be warm. Okay, in this case, and then you find the number of gas bubbles released in 5 minutes or 1 minute or 2 minutes. So the responding variable is still the same for all these experiments of photosynthesis. The responding variable is number of air or gas bubbles released in 5 minutes. Okay, and the method of handling the responding variable is still the same. Count and record the number of air or gas bubbles released in 5 minutes. And you must take the apparatus. Common mistake by students is they forgot to write down the apparatus. For responding variable, your method to handle must have the apparatus. Okay, so take note. Now, constant variable. Again, it's the same like for the other, I mean similar in some ways, some of it is similar to the other for the other factors, but it may change, of course, because here you are studying effect of temperature, so you must keep the concentration of sodium hydrogen carbonate solution constant. This will keep the concentration of carbon dioxide constant. Okay, only temperature changes. So method to fix the variable is fix the concentration of sodium hydrogen carbonate solution at 0.2%. Okay, or it could be some other value. So must state the value of the sodium hydrogen carbonate solution. Okay. Now, what is the effect of temperature on the rate of photosynthesis? So here is the graph of rate of photosynthesis against temperature. Okay, just for the theory part. And the optimum temperature for plants is 25 to 30 degrees Celsius around this region. All right. And this is when the rate of photosynthesis is the highest. Then at very high temperatures, the heat will denature the enzymes involved in photosynthesis, and the rate will decrease back to 0. Okay. Now, there is another factor that can affect the rate of photosynthesis, and that is the different light colors. Okay, so investigate the effects of different light colors on the rate of photosynthesis. So explain very briefly. Manipulated variable here is that we want to change the color of the light. So you can use a different colored cellophane paper or plastic filters, okay, or filters made of plastic, which is more durable, such as blue, red, blue, and green filters to cover the boiling tube or the beaker containing the Hydrilla species. Okay, now you can see here, the whole boiling tube will be wrapped with the plastic filter, so that the light now is shining from the top. So it will have to pass through the plastic filter. Some of the plastic filters are made from the tougher plastic. This one is for blue color, and this is for green color. Okay, now the light bulb is shining from the top down to the boiling tube. Okay, now in the boiling tube, your constant variable will be the concentration of the hydrogen carbonate solution. Maybe you fix it at 0.2%. Okay, then the responding variable remains the same as for the other experiments. Count and record the number of air or gas bubbles that are released in 5 minutes or 1 minute or 2 minutes using a stopwatch. Okay, count, not measure, count and record, because you can do counting. Count and record the RV, write down the complete RV by using a stopwatch. If you just count the number of air bubbles released by using stopwatch, wrong. You must state the time. Okay, 1 minute. So complete answer. Write complete answers. Constant variable is the concentration of the sodium hydrogen carbonate solution, and you can fix the concentration of the sodium hydrogen carbonate solution at 0.2%. That is your method to handle the constant variable. Okay, always have a value there. That's certain to get the mark for that. Okay, now method B. Now this one is shown on a lot of YouTube channels and becoming more and more popular. Okay, how to investigate effect of different light colors on rate of photosynthesis. There's this one method which you can also see on YouTube. First of all, they get the leaf disc from a leaf. Okay, from a leaf like a normal leaf okay, from plants, and they punch the hole to make the leaf disc, small leaf disc. Okay, and then they have a method to expel the excess air from the leaf spaces, spaces in the intercellular spaces in the leaf. So all the leaves without the air will sink down to the bottom at first. Okay, then they put the filter at the top here. Now this is from one experiment in the YouTube, right? And so the light has to shine through the filter. Okay, so in this case, red light will enter the beaker here. All right. Now all the leaf discs were actually sank to the bottom already. But when photosynthesis occurs, bubbles will be created. Bubbles of oxygen will stick onto the leaves. It will be released from the leaves, but it will stick to the leaf disc and make the leaf disc buoyant, and the leaf disc will float up to the top. This shows that when you float to the top, means that photosynthesis has occurred and the air bubbles or oxygen bubbles have been released. Okay, and it float to the top. So our responding variable in this case is to measure the time taken for all the lifties to float up. Okay, maybe 8 lifties to float up due to the oxygen bubbles on them. When all the lifties, 8 lifties have floated up, that means the reaction is completed and you note the time. Okay, and we use 0.2% sodium hydrogen carbonate solution to provide the carbon dioxide solution for photosynthesis. So in this case, your MV is again the different color of light, okay, that passes through the filter, red, blue, and green. And then your RV is no longer the bubbles, counting the bubbles coming up. It's the time taken for all the 8 lifties to float to the surface. Okay, and then your constant variable is the same here, just like for this experiment. Concentration of sodium hydrogen carbonate must be kept constant to keep constant the concentration of carbon dioxide. So fix the concentration of sodium hydrogen carbonate at 0.2%. Okay, there must be a value. Now, so I want to discuss the results for the experiment to study effects of different light colors. Okay, because this can be asked in the prediction question, or in the classification question. Now, results. We look at the first experiment where you put a filter over the boiling tube. You'll see that in red light, the red light is the best. The number of gas bubbles produced when you count it is the most. Okay, followed by blue light and green light. Okay, counting the gas bubbles released from the Hydrilla plant, right? And the reason or inference is that a lot of the red light or blue light is absorbed by the chlorophyll pigments, so they can be used. The energy from red and blue light can be used for photosynthesis, right? Now, some blue light is also absorbed by carotenoid pigments and transferred to chlorophyll. This is just an extra point, okay? Thus, red and blue light provide a lot of energy to excite electrons in the light-dependent reaction during photosynthesis, and they are also used for the splitting of the water molecules in photolysis, okay? They are used in the photolysis of water. Thus, the rate of photosynthesis in plants is the highest in the red and blue light, and more oxygen gas or more oxygen bubbles are released. Okay, because the red and blue light are absorbed and used for the photolysis of water. Okay, then the rate of photosynthesis increases. But what about the green light? Chlorophyll. Now, inference for green light is like that. Chlorophyll does not absorb the green wavelength of light. That green wavelength is reflected from the plant, as seen in this picture here. Okay, red light and blue light are absorbed, so the energy can be used. The energy from these two lights can be used. But the green light is reflected. That's why the leaf is green. It's seen as green. So the green wavelength is reflected from the plant. Therefore, the energy from the green wavelength is not used. From the green rays are not used. Okay, thus the rate of photosynthesis is lower in green light. Okay, higher in blue and red light, and the number of air bubbles or gas bubbles released is more for red light, followed by blue light, and lastly, green light, very little. Very few bubbles are released. Okay, during photosynthesis. Right, that's all for this lesson. Thanks for viewing, and I hope that you have learned something from this and prepared yourself better for the coming exams. Okay, so all the best to you, and may you be successful in this experiment, this exam, and stay calm and study. Till we meet again, goodbye.